So I think we can start. Hi everyone, it's my pleasure to introduce today's speaker. His name is Csaba Szabó and he is coming from the agricultural campus. He is the head of the Animal Nutrition and Physiology department and also the leader of the horse breeder training course. Csaba conducts his research and projects related to farm animal nutrition, and I think today he will present some of his latest results.
Yeah, thank you very much for the opportunity and welcome to this seminar. Yeah, I will not really present my results. I will show you some recent publications, but all the let's say thoughts are a little bit on a general basis, let's say, because since you are not really familiar or just in partly touched by the animal nutrition or animal nutrition science, then I'd like to take the opportunity to just give a kind of overview that what is all about that. So, well, nowadays there are a number of, maybe a little bit back just to let you to see it. There is a lot of challenges regarding to animal farming or farm animal production, and of course one of the main driving force is that there is a growing population on Earth still, but it's also true that according to some projections there will be a maximum, let's say, so it's not going to increase continuously. But still, since in many countries, and especially in the more populated countries, the living standards are increasing, and actually it is a kind of aim to increase the living standards, then there is a desire to have more good quality and more safe food. And this also drives a need for animal products. I have seen a FAO statistics which show that in the developed countries there is a higher consumption of animal products. So this is behind this idea that if the living standard is increasing, there is an increased need for animal products. We also have the challenge that the area of arable land is decreasing. You don't have to go very far, you can just look around in Debrecen. There is a new BMW factory, and of course it's a green construction or greenfield construction, so which means that the arable land again lost. We are building up new roads between cities, new highways, everything. Again, we are losing arable lands, and I think there is a kind of similar tendency in most of the part of the world. So this is also a kind of challenge. We should mention also the environment. Yeah, animal husbandry, of course, poses some risk to the environment. If the manure especially is not treated well, but it also depends on the country where we are. Because here in Hungary, like there is 93 or something like 1,000 square kilometer of land, and we have something like, with optimistic, let's say, estimation like 2.7 billion pigs, and compare it with the Netherlands, 1/3 of the territory, 6 times more pigs, and let's say a hill above 60 meters of the sea level called a mountain. So that's the big difference. So it also makes of course quite a bit of different approaches and view of perspective. And then of course we should mention the climate change as well. So you will see that also all research, or most of all research, let's say, some extent related to the climate change, and this is already here, and we don't know that how far going to go with these all political changes like Trump, who believes not in climate change. Just to remember that when I was still studying at the high school, one weather forecaster was fired in Hungary because he said that tornadoes will appear in Hungary. And he said, no, it's 3 or 4 years it happened, and it destroyed in the nearby area some houses. So it's certainly here, and we see that even in my life, I see that we don't have real winters anymore. So I mean, I always used to frighten the foreign students that we used to have -20, -23, in the winter. It's really freezing, but it's already a fairy tale nowadays. So yeah, it's certainly changing. It's certainly changing. And then there is many other things we still don't know yet. It came up and we have to face it. Maybe one more, which is not listed here, the development of artificial meats. Yeah, and University of Debrecen also active in that field, of course. In some extent, it's needed for the longer space travel and everything. But in the other hand, this also a kind of challenge to animal farming, how to cope with the situation and what to do, how to adapt to these things. So of course, if we go back to the first point, like the growing population and the growing need, actually it's of course the first thing is that we continuously trying to improve the productivity of all animals. And actually the next 3 slides will give you some kind of idea about that. I just recently found this image on Facebook, so I took it. And but it really shows that these are broiler chickens, and you see that in 1957, even the chicks were smaller. Nowadays, like 44 gram, and then back in that time it was 34 gram. But if we look at the final weight, 56 days. Those chicks in 1957 was less than 1 kilogram weight, and nowadays over 4 kilogram. So actually the normal slaughter weight, which is let's say around 2 kilogram, they reach it like 5.5 weeks of age. So they are really young, they are really fast growing animals. And also there is another example on layers. This is a picture used in my slides. What you have to see that the age at the end of the laying cycle is like 70 weeks of age, but nowadays the modern genotypes, even the Hungarian one, the Tetra SL, can produce up to 80 weeks of age. And then the more developed ones, the long life hybrids, can produce eggs up to 100 weeks of age. So the productivity is increased. Yeah, maybe it's similar because they reach like 95% of intensity at the peak production, and there is a certain decline, of course. And then the next one is for dairy cattle. This is showing the changing of the calving interval of the cows in Hungary. And of course, the smaller number is the better, the bigger one is the worse figure. And what you can see here is that in the 1980s, 1990s, started, let's say, a steady increase, and we reached like almost or over 440 days. What happened? This is the period before we had the Simmental type cattle, the Hungarian spotted, magyar tarka in Hungarian cattle. And then of course, this time the dairy cows was milked by hand, and also the production was like 3,000 to 4,000 kilogram per lactation period, which is rather small. So there was a need for more milk, and of course with the hand milking it was not further possible to increase the farm size. So it was not possible to manage larger herds. So what happened, it is introduced or appeared the machine milking, but for that, of course, a more uniform teeth was needed. And because this Simmental type cows had a thick, narrow, this direction, everything, so it was not square, not uniform, and so it was not suitable for machine milking. And of course the production again. So what happened? And in the US the Holstein Friesian cows, the breed has been developed. So we took the bulls and we started to cross them with the Hungarian Simmental. So we started the overcrossing. And of course the genetic improvement was much faster than our ability to learn how to feed the cows. So you see that, and why this calving interval is interesting, because every problem accumulated in that one, in that figure. So the nutrition, management, everything. And this is why it really shows very well that it took like, maybe it's like 20 years or 25 years to learn, and then now there is a decline, and in 2023 it was 407 days, so we are going back to the original figure. So we have to learn how to feed those animals, and at the same time you can see that the Holstein Friesians on country average had almost 11,000 liter or kilogram milk, not much difference. Production per standard lactation, which is a 305 days lactation. So from the 3 to 4,000 there is a tremendous increase. And as I used to explain to my students, but if you look at the broilers are not ostrich, the cows are not whales, or the pigs, I don't know what. But I'm going to say with that the size of the animals are more or less the same, which means that also the digestive tract, the size, the capacity is similar, changed a little bit but similar, not changed that extent. So we had to cope also, or to serve this kind of improvement by the nutrition. And actually, always the genetic improvement the driving force, or the main driving force, to improve the nutrition of the animals, the nutrient supply of the animals, and to have better and more units, evaluation, technique, systems of the feeding value of the feedstuffs, and also the nutrient requirement of the animals. So how it all started, so after this kind of introduction, I'd like to show some slides like how it started. The first, let's say, evidence or research-based system is dating back to 1909. There was an agronomist there who suggested that the feedstuff should be evaluated compared to a middle hay. So it was just simple comparison, more or less simple comparison, that we feed hay and we feed other feedstuff, and let's see that how big amount we need to achieve the same production. So that was a very simple approach, more or less, because he had a friend called Einthoff. And Einthoff, already at that age, he thought that those nutrients which are not dissolvable in water, dilute acid, dilute alkali, and alcohol are not useful to animals. And that was, I will show you in the next slide, that was a really look forward idea, and later on it has been really a driving force to develop a system which is still used in some extent nowadays as well. And yeah, but why it was interesting? Because actually Ter wasn't really interested in animal nutrition. He was interested only in compare the feeds, because they see or they saw that from one feed the animal growing better, and from the another feed it could be the hay growing maybe well or not so well. And he didn't want to pay the same amount for the two types of feed. So he was more like on a business level. So in their publication, they did not publish any animal requirement, just comparison of the feed stuff. So, but still, that was the very first time that they put together some kind of system. And of course, later on, in Scandinavian countries, they changed the value, the comparison feed stuff, like to barley and oat mixture, or even at the end we call it a Californian system, like glucose and milk protein casein mixture. So it was very well developed. But then what I wanted to show, that in the so-called proximate analysis system, which is dating back to 1864, developed in a place called Wendel, is a small village in Germany. There was a research institute, and it's all back to that. So then they started to build up a system by which we or they were able to distinguish between the main nutrient groups. And of course, at that age, they didn't have HPLC or whatsoever, gas chromatography and other stuff. So they had to make some simplifications. Let's see, I just tell you only one example, like the protein. They analyzed various proteins, and they found that on average, proteins contain 16% of nitrogen. And as they said, okay, that's good. Let's determine nitrogen. Very good. We multiply 6.25, and then we have the protein value of the feed. Yeah, of course, there is a lot of error in that. And also today, why it is still used? Because if I say that, okay, nowadays, let's determine protein, but the next question, okay, which one? You know, there is billions of different protein structures. Because nowadays, these sophisticated machines can detect a certain molecule. And yeah, we also need to, or want to know in general, that what we know. So that was another milestone, let's say. And then another interesting thing was, even you see that is in the 18th century, to realize that some nutrients are kind of building blocks, and another ones providing energy. They started to understand energy, you know. And one of the pioneers of that was Lavoisier, who created the first, let's say, biocalorimeter. It was a very simple one. They used a trick that they did know that 80 kilocalorie of heat is needed to melt 1 kilogram of ice. So they simply prepared, let's say, a 3-layer chamber. In the outer layer there was ice with melted water or snow. In the middle one only ice, and in the inner one was the poor animal. So what they did, you can see here in the picture that the animal melted the ice and they collected it. And they simply measured the water, okay, how many water melted in 1 hour, multiplied by 80, okay, we know that how much heat it was produced. So that was a very simple one. And then comes the more complicated one, the indirect calorimetry with the respiration chamber. This is a very early one. The trick behind that is that it's a closed system. Let's say closed, okay? Because it can measure the volume and composition of the air going in and also the air sucked out. And then it is possible, of course, sometimes they need to go in for the animal to feed and everything. So they collect the feces, the urine, the products, milk and everything. And from the carbon and nitrogen turnaround, by calculation, they can calculate the amount of heat produced based on the amount of nutrients degraded during the experiment. And these type of techniques still used in many areas. These are the smaller ones, like for mice, rats, even fish, plants, because gas turnaround in plants also interesting. And also there are bigger ones. I didn't bring a picture about that for cows or for goats, so it's still used in the more developed countries. Also, there is one in Hungary, but not operating, as far as I know. So this is the respiration chamber, and then another milestone was when it was realized that okay, it's everything nice what we are determining in the lab, but it still didn't see the animal. So there is the digestion, and of course, nothing works with 100% of efficiency. So there is some loss, and the most evident loss is with the feces. So then the digestibility studies, learning about the digestibility of nutrients started, and this is a kind of end of the stage, let's say, pictures, because it started with cows, with pigs, actually in the 1990s approximately. And of course, as we did know more about the digestive physiology of the animals, we realized that the small intestine is the place of the absorption of the nutrients, which is utilized in the body. And what happens in the large intestine is a kind of error source, or most of the time, especially in case of proteins and amino acids. So then, of course, researchers try to find out a surgery technique, how to be able to collect the digester at the end of the small intestine, which is called the ileum. So that was the ileal digestibility, and for that, different cannulation techniques has been developed, and this is pictures of one we did in the past at my previous workplace here in Debrecen. They did not do like that. In case of roosters, because it's more difficult, very tiny, small diameter of intestine. So, and what is the phenomenon of birds, that they are excreting the urine and the feces together through the cloaca. And for that reason, it is not possible to determine the fecal digestibility or let's say the ileal digestibility. And for that reason, it is only possible with some cannulation techniques. You see a picture about here, and then the rooster equipped with the different bags. So like here in the bigger one, you see some whitish stuff. So if you have seen excreta in the backyard from the chickens, you always see some greeny thing and then white stripes on it. The white stripe is the uric acid. This is from the urine, because as it goes to the air, then it precipitating and gives the whitish color, and the greenish one is the manure actually, and is the feces actually. So and the small bag through this cannula collects the feces, and through the cloaca we collected the urine. That's why you see two bags here, and you see that this bird is very nice, shiny, red colors, very eager about the outcome of the research. You can see it here on the eyes of this bird. And then, of course, when we are examining an animal, and also in our research many times we are collecting tissue samples, but we need to sacrifice the animal for that. So one important area of animal nutrition research is to use non-invasive techniques, which means that we don't need to kill the animal, and we are able to study the same animal several times. And one of the earliest technique, we can say one of it, because maybe the earliest one was the ultrasound technique, just to measure the back fat thickness, for instance, and later on some B-mode ultrasound machines has been produced, which were able to produce some cross-section, some image, but the penetration was only like 8-10 centimeters, so it's not really deep. And then appeared the computer tomograph, CT, and actually in the 1980, maybe 81, I think, Norwegian researchers, led by Skierwold, managed to obtain one second-hand CT machine from a hospital and took it to the animal science department, and they started to study animals. Sheep, pigs, fish, what they found. They scanned everything. And then, of course, in the meantime, also the CT machines go through development. So nowadays there are so-called spiral CTs, which means that they go through the animal or the man, and they can make many cross-sectional images. And as you can see here on a grain scale, you can visualize it. And then, of course, if we are making equidistant images, we are able to, let's say, calculate the fat and the muscle or bone volume in the tissues. And based on that, there is a possibility to build up a curve like we did it in my PhD back in Kaposvár. So you can see some figure about that here. But not only, but this doesn't end with the computer tomograph. There is the magnetic resonance imaging, MRI techniques, which are more detailed in the soft tissues. Also, there is a positron emission tomography with the light isotopes. We are able to track certain molecules in the body. Also, there is the newer techniques using phenomenon of these cyclotrons to detect again very small molecules in the body. They are small yet, so I mean it's not like we can put a pig inside, as you can see here. But still, this is an area to further development. So, at the end, of course, as you can see from this kind of outlook, nutrition or working in nutritional sciences nowadays is not anymore about just to find out that which nutrient concentration resides in the best production, highest egg production, growth rate, or whatsoever. So nowadays we are more and more going more in deep. So for that reason, if we are talking about nutritional sciences, there is a lot of interconnection with other fields of sciences like physiology, called nutritional physiology, microbiology, nutritional microbiology, immunology, nutritional immunology, molecular nutrition with the genetics, molecular biology, chemistry, feed analytics, extremely important, and but all of them extremely important. Technology, feeding and feed production technology. Nowadays, we can calculate a formula on paper, but still needs to be produced. And actually, for the final quality of the feed, the technology, the production technology can add so much nowadays, can make possible things. I can put, let's say, 6% oil in the formula on paper, but this is not possible to pellet. It's not possible to be pelleted, it falls apart. So how technology can help, we put only 3% in the mixture and the rest 3 on the surface. And that makes possible what was not possible before. And mathematical modeling is also very important. And at the end, we see this precision feeding. So this is a kind of new trend, or new trend, we can say it. Because of course, we need to be more productive at the end. And there are other problems in agriculture, in animal agriculture as well, that we are in lack of proper workforce, human workforce. And in Hungarian there is a saying that the farmer eyes fattening the pig, gazda szeme ízlalja a disznót, but we don't have anymore the farmer's eye. We need to increase the number of animals looked after by one person, so they don't have time. So when I had only a short time and I walked in front of the barn and looked at the pig, okay, yeah. So, but there was one lying pig. So if I don't stay there, I don't know what happened. Dead, sleeping very well, or maybe it's lame, and so yeah, you need to spend time over there, and we don't have that time nowadays. So for that reason, also different kind of sensors, the development of sensors, like there is a possibility to analyze the sound, the cough, how the animal cough. Because then with the proper, of course, justification, you can set a kind of alert that they have maybe an infection or something like that. So normally, of course, temperature, air movement, dust level, ammonia level. So there are so many things, or just like with video analysis, how the animals are spread in the barn. It also gives information, which is for a farmer, it's obvious. Okay, you see the pigs lying, it's spread, okay, everything fine. Everybody's together, oh, it's cold. Or there is a too strong air movement. So there is so many things, that's why we need also technology as a development, and at the end we are trying to make it more precise, the feeding. Of course, if we think like a little bit like a science fiction, let's say, very precise feeding would be then if we could have online or up to the moment information about the physiological status of the animal, and then we could say that okay, in the next 2 minutes you will need a little bit more lysine because the farmer or the worker came in and kicked your ass, so then you are in stress and you need some maybe little bit more vitamin E. So of course this is too futuristic at the moment, but actually we still can do a lot of things so far. And then, yeah, in the next part, I will just show you some examples of our recent research and publications. This is a review, and this is just really shows that we applied in the subsequent experiment different antioxidant treatments in the feed of the pigs, and then we examined morphological changes in the intestine, also the antioxidant system changes. So, and this was a review. It's really worth to do. I encourage everybody to do reviews. This dates back to 2021. I just checked, it already has 25 citations. We did one in the next year, 26, and we did one in 2023. It already has 17. So in your future, it's a little bit off topic, but for the PhD students, these type of publications, if you need to have a really good structure and organized review, then it can bring you citations and helps in your academic career. So this is the that was the heat stress experiment. I mentioned to you different kind of antioxidant supplementations like vitamin E, selenium, zinc, vitamin C we applied, and of course we also studying blood as a major source of or carrier of metabolites, and of course then we can have some idea, some information out of that. And then of course the intestinal morphology that was presented in Animal Science Days in Austria, and Prigi also took part in this, and he was presenting it in Hungary in Keszthely before. So it's also, let's say, what we, and this is James, my PhD student, and actually he got the postdoctoral fellowship as well. One of our project with James was to test the different methionine sources during in ovo feeding of layer genotypes. Actually, in ovo application of anything is coming from the prevention of disease or from the vaccination, and with the development of broilers, it became evident that for those fast growing birds, maybe the eggs doesn't contain all the nutrients in sufficient amount to be really viable and having a good start at the beginning. Because just think about it, there is an egg, and we can fry it and eat it. And in the other hand, it contains every nutrient and every information to become a living organism. And this is fascinating. So, and the first experiments or first trials started to inject different kind of energy sources to the egg to make more viable the chicks after hatching. And most of the experiments have been carried out with broiler genotypes. So then came the idea that, okay, and what about, let's say, layer genotypes? And since at the university we have some traditional breeds, then we included the Hungarian partridge color genotype as well, and we really see nice differences between the genotypes and also due to the injection of different methionine sources. And this is also a publication from the same project. Methionine has also a role in the antioxidant systems, so that was some genotype differences which you can see, and we were able to publish it in a very good journal. Animal Nutrition is like around 6 impact factor. So that's really, it was a good achievement. Yeah, I'm really proud of that. And then one experiment from Brigi, and this is a kind of over connection for the last part, and last thing I'd like to call your attention is that was a capsaicin supplementation, and it all nice results. And here you can see the feed, and a little bit it can be seen that there is some more reddish color of this capsaicin supplemented feed, and the supplementation was like 80 milligram per kilogram. And if we go further, somebody is also here, so I used his table from the last paper just submitted, where we tested different kind of amino acid supplementation levels in the feed, methionine and leucine, and we used plus 11 gram per 10 kilogram feed or 18 gram 10 kilogram feed, so that was a really small amount. And why I'm showing that to you and why I'm saying that, and also the subtitle of my presentation was to farm, from barn to barn, because of course everything started in the barn. And at the end, when we are curious about the effect of nutrients, bioactive compounds in a very small amount, it all goes back to the barn. Because we can do any nice, high-tech laboratory analysis. If the feed is not homogeneous, and if the animal experiment went well, then you are never know what you are measuring. If there is a difference, why is there? If there is no difference, why no difference? So just to see the challenge and to show you one example, this is happened recently, and this change, it is also relates back to the experiment of Briggy, because that was the first time when we developed the idea, let's say. Because when you have a very small amount, like in case of Doha, there was a corticosterone, was able to dissolve in alcohol and then further, let's say, increase the volume by water. And we sprayed, we increased the volume to 100 milliliter, and then we sprayed on 5 kilogram feed, because this tray, used also in the experiment of Briggy, can handle about 5 kilogram of feed. Finally, we needed here 8 kilogram per treatment. So what we did, that we put the 5 kilo feed, flat it, and then started to spray from outside to inside. Then mix by hand, flat it again, spray. Because it's so small amount that if it is a powder, then it's almost impossible to distribute it evenly. And think about that these birds was eating like, I think she was calculating like 60-70 gram of feed per day, so it's a really small amount. And then there was another treatment, creatine monohydrate, 1.9 gram per kilogram feed, which was not dissolving in anything. So there was no other choice but mix it in the feed. So what we did is that we sieved the feed. We sieved the feed first with this larger siever, and then we had let's say a bigger particle size, and we used that for spraying the dissolved material. And then the fine part was used to mix this amount. But how we did it? We know that like for the total amount we used 15 gram. And this is actually a new approach what I developed. That I said, okay, take let's say a kind of bowl, measure the 15 gram, put 15 gram of this fine material, mix it, stir it. Then okay, it became 30 gram, then put again 30 gram. And then we increased that amount until we reached the whole amount of the fine material. And during the acclimatization period, it turned out that the birds do not eat the really fine powder of the feed. So, before we did the mixing, we still, this already sieved part has been sieved further with a smaller sieve to take out the really, let's say, the dust. And then we made this approach, and then at the end we mixed those two type of parts in the feed mixer. Okay, this is a concrete mixer, yeah, but it works very fine. 4 minutes, so and then it's everything was homogenized, and according to the preliminary results, it really worked, because in one of the treatments it was expected that the feed intake and the growth rate of the birds will drop, and it happened. So this is how we know it. Because the problem is that how to check it back in the lab. Of course, it's principally possible, but you know, this type of compounds, I mean, the technique is not very developed. So I mean, there is a higher lab error. So we never can be sure. Even I have to say that in amino acid analysis, we did an experiment and we made a check of the amino acids in the feed, and in such an amino acid, of which was not supplemented in the feed, so we did not add any extra. Everything came from the raw materials. There was a significant difference between the amino acid content. And it says that, yes, something not perfectly good with the lab analysis, or it is like that, but then we still have to more rely on our, let's say, mixing efforts. So yeah, that was the aim of my let's say presentation today. That first give you a kind of idea that how animal nutrition started, and what is for, how much how intensively we need to work together. So as you have seen, there's different areas: immunology, gene expression, morphology, whatsoever. Nowadays, there is no single person to know everything in detail about that. So, which means that we need to cooperate. And the last message is that everything starts in the barn. And it's very important. And with the feeding and the feed mixing. Okay, thank you very much for your attention. Then, since I was expecting that some foreign students will be here, I put these slides because we are approaching Easter, and you know that in Hungary there is some traditions. One of them is that the farriers who are shoeing the horses, just to challenge themselves and the others, was putting little shoes and also different other motifs on the eggs, nailing on the egg. So that's one interesting story maybe to you, and also the decoration of the Easter eggs, the boiled eggs on different ways, because of course on Easter Monday the boys have to go to the girls who are the flowers, and they need to water them. Okay, in the past they take a bucket of water and really pour it on the girls, and in return they had to give some nicely decorated maybe red eggs, and that was a kind of reward for that. Nowadays boys maybe using some perfumes. Yeah, and the ladies are hiding because they get the smell from every single person. Okay, thank you.
So thank you, Csaba, for this nice presentation. We have time for some questions.
I have one.
Yeah, sure, of course.
Since you are talking about the evolution of feed and animal nutrition, nowadays we are in the stage of when we are giving the feed, it's based on age or based on the growing periods. Is there any chance in the future that it will be specific to the sex, like females different?
Yeah, sure, it's already. There is already many research regarding that, so-called split sex feeding, because of course the production, the growth, maybe it's growth, of course we're talking about growth, so the females and the male animals' growth rate are different, and of course it means that there is a different nutrient need. So theoretically and also by experiments it is proved that it works. Practically, you certainly need a farm size to do it, and of course you need to have a farm equipped to be able to do it. So that's it. So it's the technical feasibility depends on the economical circumstances. So we have knowledge about it, and yeah, we recognize it already. There are many research regarding to that how different is the nutrient requirements of the different genders. So yeah. Well,
Another question?
yes, please.
My question goes back to the challenges, the first slide that you showed. And I'm just curious about your views on insects as alternative nutrients or nutrient source, or even like feeding humans or animals. Is that a viable future or not necessarily?
Yep, certainly. I mean, it's. I do remember that I found one publication dating back to 1952 when they analyzed different insect proteins, insect meals, and even they had a short trial with pigs. So since then we know that they are contains good protein in a variable amount, of course, depending on the species, stage of development and everything. And nowadays, of course, everybody, or let's say, even in Hungary here, there was already 3 or 4 kind of protein strategy, and it all goes back to the fact that we are using so much solvent extracted soybean meal, which is imported mainly from Brasilia and also some other countries where the soybean grows very well, is produced. And we have quite a dependence on that. So there was some governmental programs, let's say, to replace this imported soybean meal with a locally produced cheap protein sources. So well, from that only the locally produced is true. Usually they are not cheap, not cheaper, because the soybean meal has a very determined nutrient content, protein content. It's which comes in Hungary, it's like 44%. We know the amino acid composition, and we can use it. There is not much limitation in incorporation rate, so it's good. And then the other protein sources produced locally, like pea or other legumes, seeds, are having lower protein quality, some anti-nutritional factors, lower productivity, and at the end, more expensive. So, and to this role is really connecting the insect protein, which now it is allowed by the European Union by law. So it is possible there is 7 insects which can be used for that. And of course, there are technologies and even companies who are producing this type of meals. But at the moment, it is like in the scaling phase, which means that we know that it is good, we know that it is work, we know that how it can be produced, but we have to learn how to produce in a large quantity. Because at the moment, the price of these insect meals are very high, so it is not worth to incorporate into the feed of pigs or poultry. Can be used in some special aquaculture feeds or bait production or whatever. Even I was surprised that even the insect oil was used in transformators, which comes the electricity, and there is a transformator from the high voltage as an oil in that. It was so interesting to me. So at the moment I see it like that. I read the news a couple of months ago that even in Hungary a big insect factory is built up near Budapest. So people are investing or this future direction. Even there is a lot of research regarding to insect proteins. At the moment, from let's say regulation side, they are farm animals. So because when it the first let's say news or idea came about insect protein, everybody was thinking, oh, they will eat every waste and everything, you know. But they are farm animals by regulation, so it means that they can eat only farm animal feedstuffs. So like corn gluten feed, byproduct of the starch industry, is used as a growing material. So cannot be used like kitchen waste or manure or something like that to grow these insects. So this is how I see it. So it's certainly one direction of the future. Here in Hungary we will be never able to completely independent from the solvent extracted soybean. Our land, arable land is not sufficient to produce all the protein sources we need, and also these kind of alternatives will not provide 100% of substitution. So partly yes, we'll be able to and we will use it. Everything depends on the economical situation at the end.
I have three questions. One is just came to my mind when you mentioned this. That was a big scandal, like I don't know, in the public when they forbid the kitchen waste to give it to pigs, and I didn't follow the regulations, so it is still forbidden, and
Yes.
restaurants just still throw it away.
It's a kind of dangerous waste, so they have to have a contract with some company. It can be like composted or something like
It cannot be given.
Cannot be fed to the.
It's kind of weird because if it was good for humans, how come it's not good for?
Yeah, it's a quite an obvious logic, and you are completely right. I mean, but then there is still a possibility to grow some microbes, and you don't know that how it is treated, how quickly gets in front of the animals. And from another point of view, it's the nutrient content is highly variable.
Mm.
So of course, if it is, let's say, dirt cheap, most probably you pay nothing for it, then of course it works because they grow slowly or more slower, but still, you know, no feeding cost, then it works it. But in terms of intensive production, it's not a good feed source because it's very variable. It has a very variable nutrient content.
Okay, the other question I had is that how does environmental consideration weigh into the animal nutrition? So, for example, producing or giving nutrients to animals that produce less nitrogen, for example, in manure, or for example, that cows produce less methane or something. Apart from the production and profitability and so the nutrition part, how does this weigh into this equation, if at all?
Well, if we are talking about animal agriculture, the two main pollutants is nitrogen and phosphorus.
Mm-hmm.
Okay, nitrogen is of course coming from the protein degradation, synthesis, and amino acid degradation in the liver. So how can we improve that? Increase the digestible protein content of the feed, increase the digestible amino acid content of the feed. So it can be some technology or purposely selected feedstuffs, using enzymes in the feed, using the industrially produced synthetic amino acids because their digestibility is practically 100%. So these things and even research shows that compared to the recommendations, we can reduce the crude protein content of the feed like 2 to 3% if we add more quantity and type of synthetic amino acid. Even I see research when for piglets they prepared a feed without any protein source, like no soybean, no skim milk, or milk powder whatsoever, only, let's say, grains, wheat, barley, and corn with amino acid supplementation, and the growth of the piglets wasn't really much worse than the normally fed animals. So this is also one direction. So this is how it can work. Also, yeah. So improving the digestibility and increasing the digestible part of the protein, and then of course less is non-digestive. And in the other hand, we're using this so-called ideal protein concept. So if we, let's say, accept that the proteins are species specific, then of course there is an optimal amino acid ratio, which if we serve it in the feed, then there is no waste. Of course, this is a very ideal situation, but if we increase that, and that's where the synthetic amino acids come in again, because of course the feedstuffs itself having a variable amount of amino acids, and there is always some which is in surplus, and for that reason, and there is some limiting ones. Lysine is the most limiting amino acid, so that's why the first factory in Hungary producing amino acid was a lysine factory nearby Csaba, was where the factory was built up. So that was the first one. Because then if this one limiting, then even the other ones. Just think about, I used to explain it to my students, that there is the BMW factory and you would like to build up, let's say, 10 cars, and you have the 10 engine, but you have only 20 tires. And you need 4 for one car, so you can assemble only 5 cars. So this is with the proteins as well. If one amino acid limiting the protein formation, then the other ones will not be utilized. So this is how this industry and the synthetic amino acids can happen in that one. Another thing is the phosphorus. There's a problem that up to 50% of the phosphorus in the plant materials are in the form of phytic acid. This is a kind of ring-shaped molecule with 6 phosphorus. We don't have, or the animals doesn't have enzyme to break it down. So which means that in the feed, to achieve the proper phosphorus supply of the animals, we need to add more supplementation, which is in Hungary most of the times monocalcium phosphate. So if we are using like phytase enzyme added to the feed, then we can improve, or we can make the animal capable of break down these phytate acids, and then we don't have to add that much of monocalcium phosphate as well. And at the end there will be lower excretion, 30 to 40% we can decrease the excretion, and also similar magnitude in the nitrogen. So because this is the two main pollutant, because it can go to the surface water and then could cause overgrowth of eutrophization of the waters, and also oxygen in the water. And so on. Okay. Third question?
I'm sorry, we have to leave the...
Yeah, I think so.
Because there is a press here, but we can continue the discussion at the...
Oh, okay, sorry.
Thank you, Csaba.
Sorry, yeah, it's still more